Everyone, and welcome to Lecture 6. We're going to talk about vision today. We're going to start in the retina, move through the thalamus very quickly, and end up in the occipital lobes, where we will make sense of this information. So the retina is where it all begins. This contains light-sensing photoreceptors that will turn light energy into electrical and chemical signaling that can then be relayed to our nervous system. Now, when we talk about light, usually we're referring to visible light, which is really just a very small portion of the electromagnetic spectrum. So electromagnetic energy is energy that's going to travel with wave-like properties, and depending on the wavelength or the frequency of the energy, that's going to determine what type of electromagnetic energy we're talking about here. It could be very high energy, gamma rays that are able to actually damage DNA. They have so much energy in them. Or they could be longer radio waves, which can just pass through our body without causing any harm. Light, visible light that is going to range between about 400 and 700 nanometers. The shortest wavelength or the highest energy there is going to be the bluest light, and the longest wavelength, lowest energy, will be red light. So since light travels as a wave, it's going to of course have amplitude as well as wavelength. Wavelength is what determines the color, as we just discussed. Amplitude will determine essentially how bright the light is. So if the photon, the little light particle, is traveling with a large amplitude, then it's going to appear brighter to us. Now, typically we're not viewing lasers, so there will be multiple wavelengths of visible light in whatever we're looking at, and so that's how we get a mix of different colors with different saturations. If you were to mix green and red light in equal parts, you'd have light that appears yellow. If you were to mix all the wavelengths together, you would have white light. And so then in this mixture, if one wavelength is present a little more than others, for example, if you have some additional longer wavelength light, but there's still all wavelengths present, you'll have a barely red, but still very much pale light. But this is not a physics class. We'll move on here. You should just be aware that light has different frequencies, and the different frequencies are going to determine the amount of energy in the light, as well as what we perceive as color. Of course, the light has no color until we create it in our brains. Now, if we're going to take in visual information, we have to be able to move our eyes. Our eyes are where the magic happens. And as we move our eyes around, typically these movements are jerky, what we call saccadic movement. So there are saccades or saccades, depending on how you like to pronounce things. These are the rapid movements where our eyes dart from one location to another. Hopefully the video on the top is now playing, and you can see these jerky movements left and right. Her eyes will almost instantaneously move between their target. As you look around the room to find some object of interest, your movements are ballistic. They're rapid. Those are the saccades. When you don't have an object that you're fixed on but you're looking for it, eye movements are instantaneous. In addition to the saccadic movements, we also have smooth movements. There's really only two types. Sometimes you'll see the movements broken down into 3, sometimes 4, but really there's just 2. They're either saccadic or non-saccadic. So they're either fast or they're smooth. Now the smooth pursuit, which hopefully we can see now in the middle video, now you can actually follow her eyes as she's tracking the cotton swab from left to right. She's not looking around for the cotton swab. She knows what it is. Now she's just simply following it. And you can actually watch her eyes move smoothly from left to right. So if you were to try to just scan the room right now from left to right without following anything, you're going to move from left to right with a few jumpy movements. But if you were to try to do the same thing while following the tip of your finger as you move it from left to right, you'll notice that you're now capable of smooth pursuit. So our eyes only move smoothly in order to hold an object in our in our gaze in order to keep it in our center of our visual field. This could be from left to right, as we're seeing there. We can also have vergence movements, which hopefully we can see on the bottom, where our eyes actually move in different directions. What we're looking at is convergence. As the cotton swab moves closer to her eyes, they turn inward, so they move together. So they kind of cross there in order to hold the cotton swab in the center of her visual field. As the cotton swab moves further away, we can see divergence. But what you'll notice is that these movements are smooth. They are not saccadic here, because she already knows what she's fixated on. She's not looking for anything. She's just simply following. Now we want to hold our objects of interest in the center of our visual field, because we need to receive that light input onto our retina, and we need to hold it fairly constant if we're going to make sense of it. So the job of our eyes is to focus the light, which they'll do with the frontal portion of the eye there, and then detect the light, which is what will be done in the retina. So the light's going to enter through the pupil after passing through the outer covering, the cornea there. The size of the pupil is, of course, controlled by the iris. We want to keep a relatively constant amount of light on our retina. So if you're in a dark room or a bright room, you still need to have a decent amount of light in the retina. You don't want it so bright that you can't distinguish the brightest values, and you don't want it so dim that you can't see anything. So by opening up our pupil, so by dilating the pupil or by constricting the pupil, we can let in more or less light to make sure that our retina is receiving the appropriate amount of light to make sense of what we see. As we look at different parts of the room or the world, some might be very far away, some might be very close, so we have to adjust the focus of light so that we can actually see clearly whatever we're looking at, whether it's near or far. And that's the purpose of our lens. So there are little tiny ciliary muscles that will pull on the lens. The lens is really just a little crystalline structure of proteins. The proteins are called crystalline, in fact. So this is a little stretchy. When you pull it, you change the curvature of the lens, and thus you change which part of the world is in focus on your retina. This is a process that we call visual accommodation. So once we've looked in the right spot, we've let in the correct amount of light, and we focused it on our retina, now we can start to make sense of the world. So the retina is where the magic happens. The retina is what's going to transduce our visual field. That's everywhere that we're looking at into electrical and chemical signaling. And it's going to do that using different cells that are contained in different layers of the retina. What we're going to find is that the retina is very much non-uniform. First of all, the retina has a few different layers. We can break it down into 3 different layers. There's the outer nuclear layer, the inner nuclear layer, and then the ganglion cell layer. The outer nuclear layer is what contains our photoreceptors. There are rods and there are cones. If you look on this illustration on the right, you'll see the rods and cones on top. This is the back of the retina. So light has to pass through the ganglion cell layer on the bottom, then the inner nuclear layer, and then finally hit the photoreceptors. So rods are going to respond to essentially all wavelengths of light. They're not going to distinguish different colors, but because they pick up such a wide range of light, they can respond to very dim levels of light. Cones, on the other hand, are going to respond to more specific wavelengths of light, essentially red, green, or blue, but because they're restrictive of their wavelengths, they need brighter amounts of light in order to sufficiently activate them. Once we've activated our photoreceptors, then they're going to signal to cells within the inner nuclear layer. The principal cell that we need to know about would be the bipolar cells. These are the middlemen between our photoreceptors on top and the retinal ganglion cells on the bottom. We'll see that there's 2 types of bipolar cells, more on that later, so that we can have a different degree of signaling, and this will allow color detection. Then we have within that inner nuclear layer a couple of other cells that are involved in doing some minimal processing of the visual information. Horizontal cells are going to deliver GABAergic input to the photoreceptors to help improve contrast at edges. And the amacrine cells are actually a diverse class of cells with a variety of different functions. We'll just sort of sweep them under the rug and not worry about our amacrine cells at this point. But they're going to communicate to the retinal ganglion cells. The retinal ganglion cells we will absolutely not sweep under the rug because these are the output cells from the retina. These create the optic nerve. So these are going to project from our retina to the lateral geniculate nucleus of the thalamus. All right, so these are the nerve cells that then conduct information from the retina into our central nervous system. Now the retina has this portion called the fovea. The fovea is the portion of the retina that receives input from the center of our visual field. So wherever you are looking right now, that is what's hitting your fovea. The center of your visual field is hitting your fovea. And the fovea is going to give us the greatest visual acuity. If you want to look at something and really take in the details, you look at it. You put it in the center of your visual field. You don't look kind of to the left or right or kind of away from something if you want to actually make sense of it. When you're reading, you look at the words you want to read. This is because of the difference in coupling between retinal ganglion cells and photoreceptors, as well as the difference in what types of photoreceptors are there. So the fovea only contains cones, so we can see color there, and then it's going to be one-to-one coupling of photoreceptors to retinal ganglion cells. That allows the retinal ganglion cell to respond to very fine details because there's only a single photoreceptor that can activate it. Whereas in the periphery of the retina, each retinal ganglion cell is going to receive input from multiple photoreceptors. Now this means that there's less detail because there's a larger area of the retina getting funneled into a single ganglion cell. So the ganglion cell is actually responding to a much larger portion of our visual field. But because it's doing that, dimmer levels of light are able to activate the retinal ganglion cell because it's not reliant on just one photoreceptor to activate it. In this cartoon, it's reliant on 6 photoreceptors. So it should be able to be activated by 6 times dimmer levels of light. This is why if you're ever trying to see a star at night, you do actually look a little bit away from it. Because if you look at it and put it in your fovea, you only have one-to-one coupling. The photoreceptors there need much higher levels of light in order to be activated. But if you look a little away from it, then you're putting that star not in your fovea, but in the periphery of your retina, where you have higher levels of coupling between your photoreceptors and each individual ganglion cell. So the low levels of light from that dim star are going to fall on multiple photoreceptors, and their activity will be summed and integrated into a single ganglion cell. So we sacrifice detail in our periphery in order to have a little more sensitivity to light. Now the inverted organization of our retina is odd, and it does indeed pose a small problem. It creates a blind spot. So the photoreceptors, that outer nuclear layer, that's at the very back of the retina. The retinal ganglion cells, which actually create the optic nerve, they're actually at the front of the retina. So their axons have to pierce through the retina at some point in order to project back to our brain. The point at which they pierce the retina is what we call the optic disc. This is an area that cannot contain photoreceptors because the axon is piercing through the retina in order to project back to our brain. And so in this area, which is thus devoid of photoreceptors, we see absolutely nothing. Your book has a very nice illustration of the blind spot. I do suggest that you check it out. It's kind of nifty. You have one in each eye, but you never notice it because information from the other eye generally can be used in place of that blind spot. And if it can't, if your object is too close for the other eye to cover the blind spot, then our brain just paints over it with whatever it thinks is there. It's kind of nifty. Okay, so the rods and the cones, they're able to detect light because they have what we call photopigments. Photopigments are proteins that are able to absorb light. They're a collection of retinol and opsin. Retinol is the molecule that actually absorbs light, and there are different versions of retinol that will absorb different frequencies of light because the retinol is going to be combined with opsin, and when it does that, it changes the wavelength at which the retinol will absorb depending on the opsin protein. The opsin itself is a G protein coupled receptor, so it's a metabotropic receptor, so it's going to cause some sort of intracellular signaling in response to light. Rods will express only one type of opsin, just rhodopsin. It's color insensitive, it doesn't care. So it's going to allow retinol to absorb a wide variety of wavelengths of light. Cones, on the other hand, are going to express one of three types of opsins. So there's the L opsins, which L means long wavelength. They'll absorb the red and yellow wavelengths of light. M opsins absorb the medium wavelengths, so the greens and yellows. And then there's the S opsins, which absorb the shortest wavelengths, so they'll absorb the violet or blue lights. So this is what allows cones to distinguish red, green, and blue. You'll see whenever you look at the actual frequencies that are absorbed, there is a difference in the peak absorbance. You'll see that the S cones have their peak somewhere between 400 and 500. M and L have a difference, but there's quite a bit of overlap in the actual absorbance itself. There's a difference in the max, but there's some overlap. So in order to really determine the color, we have to look at the activity of all three types of cones, and then we can make a determination of what color we're really looking at. Just one cone won't cut it. I want to touch back on our fovea real quick. So the fovea is where we actually perceive color. So that contains only cones. There are very few rods in the fovea, and there are very few cones outside of the fovea. So there's our fovea there, right in the center of the visual field. If you were to take a moment and count all of the rods and cones in the retina, which was done here back in 1935, what you'll find is that cones are at the greatest concentration in the fovea. If you look at the x-axis, you're looking at the angle away from the center of vision, so 0 right there in the center, that's your fovea, and you'll notice the cones have a nice spike right there. The rods are actually at a low point in the fovea because it's dominated by cones, but when you look in the periphery, there's a whole lot of rods, except of course for that blind spot where the optic nerve leaves the retina there. So that means that your peripheral vision, believe it or not, is absolutely colorblind. There are very few cones, and it's difficult to perceive differences in wavelengths of light. We don't see the world as gray in our peripheral vision because we have some memory of what we've looked at, and so our brain just sort of automatically paints over all the colors that it remembers being there. Okay, let's talk about how the photoreceptors actually respond to light. So we have our photopigments, and we have retinol, and we have our opsin. Here we're looking at a rod cell, so just good old rhodopsin will absorb any wavelength of light for the most part. Now remember, opsin is a G protein coupled receptor, and that G protein is what we call transducin. Okay, so we're in our rod. Light has come in. Light's going to be absorbed by the photopigment, and when it does that, it causes the G protein to then dissociate from the receptor. The G protein can then interact with its targets. Okay, so when the retinol absorbs the light, what it does is activate what we call a guanine nucleotide exchange factor, or a GEF. That is what causes transducin to leave. So it swaps the GDP with a GTP. The details don't matter so much for you folks, but for those of you biologically oriented, when the G proteins are bound with GTP, they dissociate from their receptor, and then they're free to move about the cell. They'll move around, they'll interact with their targets. In this case, it's a phosphodiesterase. That's a protein that's going to start breaking down cyclic nucleotides like cyclic GMP. Cyclic GMP or cyclic AMP are going to activate cyclic nucleotide-gated cation channels. So under normal conditions, when we're not receiving light, this cation channel is open because cyclic GMP levels are very high. If you look on the right-hand portion of this illustration, you'll see the cGMP is interacting with that cGMP-gated ion channel, allowing sodium to enter and depolarize the photoreceptor. So when we receive light, transducin is activated. It activates that phosphodiesterase, PDE there. All right, that's what we're seeing on the right-hand side. Cyclic GMP gets broken down. So cyclic GMP levels drop, and as such, the sodium permeability drops. So we experience hyperpolarization in response to light. This might be opposite of what you expected. You might expect photoreceptors to be activated by light, but that's not the case because photoreceptors are always releasing glutamate. They don't fire action potentials, they're just always leaking glutamate out. And so when they're hit by light, they get hyperpolarization, and the response to that is to secrete less glutamate. So the bipolar cell that receives input from a photoreceptor is going to get a different amount of glutamate depending on how much light is there. The more light there is, the less glutamate. Now glutamate can have one of two responses. In this case, what we're looking at is an on-bipolar cell. So the loss of glutamate actually causes depolarization. Not every bipolar cell responds this way, but in this one, it gets turned on by the lack of glutamate. It then stimulates the retinal ganglion cell to fire a higher frequency of action potentials. This isn't always the case. All right, there are two types of bipolar cells. There's the on and there's the off. We just saw the on. Let's start with off bipolar cells here. So to remind us where we are in our retina, we've moved through the top. We're done there. That's our photoreceptors. So light causes them to hyperpolarize, and now they're releasing less glutamate. So now we're in that internuclear layer where the bipolar cells are either being turned on or off. Okay, the off bipolar cells are fairly straightforward. They express ionotropic glutamate receptors. So the kainate and AMPA receptors that we've already heard about. These are standard cation channels. Glutamate binds, opens up, there's depolarization. That means when light hits and hyperpolarizes the photoreceptor, there's less glutamate, so there's less activity of these ionotropic glutamate receptors, and thus the off bipolar cell hyperpolarizes. The on bipolar cell expresses glutamate receptors but not ionotropic glutamate receptors, instead metabotropic glutamate receptors, or mGlu-Rs. These metabotropic glutamate receptors are very similar to rhodopsin. The mGlu-Rs on the on bipolar cells are also going to activate a phosphodiesterase, the exact same type of protein that we've already mentioned. That's going to break down cyclic GMP. The cyclic GMP levels, when they drop, that means that the cyclic nucleotide gated cation channels close. Causing the cell to hyperpolarize. So, light causes the photoreceptor to hyperpolarize. As such, there's less glutamate. With less glutamate around, the mGlu-Rs are not as active. So there's less transducin activity. Thus, there's less phosphodiesterase activity. That means that cyclic GMP levels rise, and as such, the cyclic nucleotide gated cation channels are more active, so we actually experience depolarization in the absence of glutamate. That's how we have a difference between our on and off bipolar cells. Then the bipolar cells are going to release glutamate onto the retinal ganglion cells, which will act as the output of the retina. Just a quick note on horizontal cells and their function. They release GABA onto photoreceptors and hyperpolarize them. Remember, photoreceptors are always leaking glutamate, so they're always activating the horizontal cells, and so the horizontal cells are always releasing GABA. So there's this tonic signaling going on in our retina. When light hyperpolarizes a photoreceptor, that's going to reduce the amount of glutamate that they release. As such, the horizontal cells will release less GABA. Because there's less GABA release, that means that all the photoreceptors near that activated photoreceptor are going to become depolarized. Thus, they're going to increase the amount of glutamate that they release, and this provides much greater contrast. Only the photoreceptors that are being stimulated by light will be hyperpolarized. All the rest will be depolarized. This creates an even greater disparity between the level of glutamate output between the activated and the inactive photoreceptors. So when light hits a photoreceptor, not only does it hyperpolarize that photoreceptor, it also indirectly depolarizes the nearby photoreceptors that aren't being stimulated through the activity of horizontal cells. So this allows us to have greater contrast at borders of light and dark, because that'll be the areas where we have nearby photoreceptors being activated by the light region, and then other photoreceptors not active by the dark region. And then this difference gets amplified by horizontal cells. Thus far, we've only mentioned photoreceptors as being light sensitive, but there are some retinal ganglion cells that actually express a photopigment, melanopsin, and thus they are sensitive to light. So just like the other opsins, melanopsin is going to activate transducin. The retinal ganglion cells that actually express melanopsin, these are going to directly transmit light information to a region of our brain called the suprachiasmatic nucleus that helps us keep our circadian rhythms, so our sleep-wake cycles. These retinal ganglion cells don't help us make sense of the world in terms of understanding visual input. Instead, what they do is entrain us to light-dark cycles. And this is what allows even some blind people can still have the appropriate sleep-wake cycles that are synced up with daylight, even though they can't actually see. So on the right, we're looking at data from mutant mice that lack photoreceptors. So they have a mutation; they're no longer able to make rods and cones. You look at panel A on the left and panel D on the right. Panel A is wild type. This is a genetically normal mouse. You can see the dark outer nuclear layer there, which contains the rods and cones. It's noticeably absent in panel D on the right. So this is our mutant mouse. It still has the internuclear layers and the retinal ganglion cell layers, but it does not have photoreceptors. As such, it's blind. It can't see. But when you stimulate it with different levels of light to help entrain its sleep-wake cycles, you can see the amount of entrainment on the y-axis relative to the different strengths of light. So as we move from left to right, they're shining brighter lights onto the wild type or mutant mice. And as the bars get larger as they move up the y-axis, we're seeing greater degrees of entrainment, which is exactly what you'd expect if you're able to sense the light and then modify your sleep-wake cycles as a result. So even blind mice, in this case, can be entrained by light because of the retinal ganglion cells that are expressing melanopsin and are thus sensitive to light. But for the most part, retinal ganglion cells are going to be involved in detecting inputs from bipolar cells because of the activation of photoreceptors. And those retinal ganglion cells are going to send this information that they pick up to the lateral geniculate nucleus. It's a very small portion of them that project to the suprachiasmatic nucleus. And the lateral geniculate nucleus is the portion of the thalamus that relays visual information to our visual cortex. So when you look at the receptive field for an individual retinal ganglion cell, you find this kind of surprising organization that we call center-surround organization, where there's a center of their receptive field, so the portion of the visual field that they respond to, and then surrounding that there's an opposite area. So sometimes the center will stimulate the retinal ganglion cells, sometimes the periphery will. So on-center retinal ganglion cells are going to be activated when there's light in the center of their receptive field. They'll be inhibited when light enters the periphery of their visual field. Their receptive field. Off-center retinal ganglion cells are going to be turned off whenever light is in the center of their receptive field. Instead, they're going to be activated by light that's in the periphery of their receptive field. And we can see the difference in this activity on this cartoon on the right. A is just showing you the on-center field, so the white portion of the receptive field, that's the part that will turn on the retinal ganglion cell. The surrounding portions are going to turn it off. So the response then to light, if it hits the center of the dot, you can see in part B, the vertical lines are showing you the action potentials generated by the retinal ganglion cell, and you can see that there's a lot more of them when light is in the center, shown on the top, versus in the periphery, that's shown on the bottom of part B. If we get the entire center illuminated, you can see in part C that we get an even greater increase in action potential frequency, and if the entire periphery is illuminated, we get an even greater decrease in action potential frequency. Diffuse illumination doesn't really do much. Of course, they're also off-center. You can see in part F, and this activity is summarized in part G. If you compare this to part B, you can see that stimulating the center in G causes a decrease in action potential frequency, while it causes an increase in part B. Then the periphery in part G causes an increase in action potential frequency. So these different retinal ganglion cells are going to respond to light in different parts of their visual field. Now, just as there are on and off retinal ganglion cells, there are also P and M retinal ganglion cells. So let's just take a quick note of where we're at. So we've handled the retina. We're now moving out of the retina. Okay, so what we're looking at here, the left and right half of the visual field is shown in red and blue, respectively. You can see that in the primary visual cortex, shown in the back of the brain there, here we're getting different parts of the visual field. We're not getting just input from one eye or the other. We're getting input from both eyes. Okay, and there's a relay station, the lateral geniculate nucleus, or the LGN, along the way. All right, that's the level we're going to hit now. So these different types of retinal ganglion cells are going to project to different parts of the lateral geniculate nucleus. If you were to slice through the lateral geniculate nucleus, you'd see different layers. The first two layers are what we call the magnocellular layers. They have large cells. And then there's the parvocellular layers that contain smaller cells. There's another layer, the coniocellular layer, that's found between these different layers. Here we'll just focus on parvo and magnocellular. So the P cells are the retinal ganglion cells that project to the parvocellular layers with little cells. The M cells are going to project to the magnocellular layers. Now, these different types of retinal ganglion cells are going to detect different information. Okay, the P cells are going to have much smaller receptive fields, and they are going to be sensitive to color, whereas the M cells are going to have much larger receptive fields, and thus they are more sensitive to dim light. So they're going to send more information about the movement of objects rather than the actual color of them. Okay, color selectivity is going to be conveyed by our P cells here. Alright, in this cartoon we're looking at our different photoreceptors that express different opsins. We have the L opsin expressing red cone, we have the M opsin expressing green cone, and we have the S opsin expressing blue cone. These are going to be coupled to the retinal ganglion cells on the bottom through a variety of bipolar cells, either on or off. So some retinal ganglion cells will receive input from a photoreceptor via an on bipolar cell, and then a different photoreceptor by an off. If we look at the red retinal ganglion cell down there on the bottom of the cartoon on the left, you'll see that it's getting excitatory input from the red photoreceptor and inhibitory input from the green cone. So there's an on bipolar cell between the red cone, and there's an off bipolar cell between the green cone. So that means that this retinal ganglion cell is going to be stimulated by red light but inhibited by green. So only the reddest wavelengths will activate it. Anything between, like a yellow, that's going to cause equal activation and inhibition. Right next to it, if we move over one, you'll see a little green retinal ganglion cell. This is the other color opponent. It's inhibited by red and activated by green. That's because there's an off bipolar between the red cone and an on bipolar between the green cone. So only the green wavelengths, but not the reds, are going to stimulate this retinal ganglion cell. So we have red-green color opponent, then we have blue-yellow color opponent. The yellow color opponent, the yellow on is going to be stimulated by red and green, but it's going to be inhibited by blue. The blue on color opponents are going to be stimulated by blue and inhibited by red or green. So only blue wavelengths will turn on the blue on. Anything that's longer wavelength will inhibit it. Then there are other retinal ganglion cells that respond to all colors. They just sense dimness. Okay, those are going to be the M cells, which we don't really care that much about. But by doing this comparison between the amount of activity in the red, green, and blue cones, we can determine what color we're looking at. And then by taking the relative activity of all these different retinal ganglion cells, we can then start to tease apart which colors are making up the visual field. So all those color opponent retinal ganglion cells would be the P cells that project to the parvocellular layer. Now the P cells, very confusingly, are also called midget cells because they have very small receptive fields. This allows them to have much finer visual acuity, but it requires much brighter light. The M cells, which very confusingly are called parasol cells, have very large dendritic arbors, and thus they have very large receptive fields. They're going to be coupled to a lot more photoreceptors. This makes it hard for them to distinguish different wavelengths of light, but it dramatically improves their response to light. So they can respond to much dimmer levels of light. So this is why the M cells, the parasol cells there, the big ones, are going to send information more about the contrast and the dimmer signals that we're seeing in our image. The P cells are going to respond to the brighter and color sensitive portions of our visual field. So our P cells and our M cells project to the lateral geniculate nucleus. They'll go to different layers depending on what they're sending. So in the lateral geniculate nucleus, we have color information going to our parvocellular layers and the bright information going there. The dim signals are going to be sent to the magnocellular layers. And then from our LGN, we're going to project to the striate cortex. And in that striate cortex, that's our primary visual cortex. That's where we detect very specific features in our visual field. We don't make sense of the world yet. We're going to break it down into very minute details. Okay, so now we've completed our journey from the retina to the LGN, and now we are in the primary visual cortex, also called the striate cortex. So each hemisphere of the striate cortex is going to have a map of the contralateral half of the visual field. So our left striate cortex contains a map of the right visual field. You'll notice in this cartoon that some of the right visual field comes from the right eye, some of it comes from the left eye. The right hemisphere of our striate cortex receives input on the left half of the visual field. So it receives some input from the left eye, some input from the right eye. In that striate cortex, we're going to detect very specific features. We don't recognize faces here. We don't determine what object we're looking at. Instead, we break it down into very fine details. What's the orientation of the lines? Are they moving? What's the spacing between different lines? That'd be the spatial frequency. Is there a difference between what our two retinas are seeing for the same object? In other words, how close or far is it? What color am I looking at? We don't construct anything beyond that. Very fine details. Now, if you were to record the activity of neurons in the striate cortex, you'll find that most of them are going to be sensitive to the orientation of lines. That's largely what our striate cortex picks up. What's the direction of all the lines here? Most neurons are dedicated to that task. So if you look at this cartoon on the left, what they're doing is rotating a line at different angles and then showing you the unit activity of a neuron in the striate cortex. So in the middle of their recording, they're showing you when they flash the line into the visual field, and you'll see at different angles you get different degrees of activity. When the line is vertical, you get a very quick burst that's around the center of this cartoon, and as it moves further away from vertical, you get different degrees of activity. It's silent when it's horizontal. So depending on the rotation, you can get different degrees of activity. Not only does the angle matter, but also the location in the receptive field. Many of the neurons are what we call simple neurons. They have your typical surround inhibition, where only in the center of the receptive field will the line cause excitation. If it moves a little to one direction or the other, that's going to enter the inhibitory regions that surround that excitatory region. So if you look in the illustration on the right at the top, you'll see on the left, when that black line is in the center of the visual field, we get excitation, but as it moves away from that into the inhibitory regions, we now get inhibition. Complex cells don't have this surround inhibition, so they'll respond to the angle no matter where it is. And whether it's light or dark doesn't matter. They just need to see this particular angle of line, and they will be excited. Then we have hypercomplex cells, which respond to specific angles, but they contain not surround inhibition, but end inhibition. So they're there to detect the ends of lines. So only if you have a specific angle, but it stops in that receptive field. It can't follow through the entire receptive field, because then it's going to hit the excitatory and the inhibitory regions, and you'll see no activity. You'll notice in the bottom illustration that the hypercomplex cell is only excited when the line is in the correct orientation and it stops before hitting the inhibitory region. So the simple cells are there to find lines in the center of their receptive field of a certain angle. Complex cells just look for a certain angle, and then the hypercomplex cells look for an angle that ends. So they're there to detect the ends of lines. The initial experiments were done with lines, but you can just as easily excite neurons in the striate cortex by showing them sine wave grades, which we're looking at there. Sine wave gradings are showing you different bands of light that kind of smoothly transition from dark to light. Neurons respond very well to these. So they're not just detecting lines, they're actually detecting the relationship between lines, what we call the spatial frequency. So the distance between those bands in the sine wave grading gives you some idea of the amount of detail that the neurons are picking up. Some neurons respond to very wide, low-frequency information. Others respond to very high-frequency, detailed information. To give you an idea of what we're talking about, here's an image broken down into low, medium, and high-frequency components. You'll see the original image in the upper left, the low-frequency component, so where the different bands of light are separated by greater distances. You can see this pretty much approximates the original image. But the edges, the fine details, that's what we pick up in the middle and very high frequency information. So different neurons are there to respond to different frequencies of information. Those neurons that detect edges, they're going to respond to very high frequency information, and those that are there to detect general contrast are going to respond to low spatial frequency information. Now we also respond to retinal disparity. There are some neurons in V1 or the striate cortex that are going to respond when the image is hitting the same spot in both retinas, or they will respond if the image is hitting different spots in the retinas. Okay, so in this cartoon on the right, we can see that there are two eyeballs there looking at a triangle. So the point of fixation for both eyes is that triangle. That's what's going to hit the fovea, and so the neurons in V1, in the striate cortex, some of them will be active when the object's in the plane of focus. And so those neurons will tell you that you are looking at this image. You'll notice that there's a square object in front of the triangle. That's going to hit different spots of the retina. You'll notice that it's going to go to the left of the fovea in the left eye and to the right of the fovea in the right eye. So here's where we have what we call retinal disparity. the two retinas are seeing different things because they're in different locations on our head. This retinal disparity is going to tell us that this object is not in the same plane of focus. If it's moving to the left on the left eye and to the right in the right eye, then it's in front of the triangle. And the reverse would be true if it were behind it. But there are certain neurons that will fire at a greater frequency whenever an object is in different locations on the retina. And that's what signals to us that it's not in the same plane of focus. And that's what allows us to perceive depth. Okay, so by sensing retinal disparity, we know where roughly is this object. Is it in the plane of focus, or is it in front of or behind wherever I'm looking? And then, of course, we also have to determine color. There are color-sensitive neurons found in what we call blobs. If you were to stain the striate cortex with a dye that picks up cytochrome oxidase, this is an enzyme in the mitochondria, you'll find that there are certain regions that have very high levels of cytochrome oxidase. So if you look in the upper right hand, that's exactly what a cytochrome oxidase stain would look like. We have V1, which is where we're hanging out right now. V2, we'll get to that in a moment. V1, you'll see little tiny dots. Those are the blobs in the striate cortex. They have a diameter of about 200 nanometers or so, and you'll find them in the second and third and fifth and sixth layers of the cortex. Remember, the cortex has 6 layers. So different layers contain the blobs. They're not found in every single layer. Those blobs contain the color-sensitive neurons, so they receive input from the parvocellular layers as well as the coniocellular layers. So red and green comes from parvocellular, blue information comes from coniocellular. They're not receiving input from the magnocellular layer. They don't care about that. These are the color-sensitive neurons. Now, if we were to put all of this together, we'd find that our striate cortex contains about 2,500 different modules. We call the striate cortex modular. It's made of these repeating units that contain orientation-sensitive neurons, the neurons that respond to retinal disparity, and the color-sensitive neurons found in the blobs. So there's the blobs that detect color, and then there's the interblobs, or the space between blobs, that detect orientation. Now within each module, you're going to have about 150,000 neurons. That means that each individual module can have about 2 to the 150,000th power different patterns of activity. That's nearly infinite. It's a number that's so large you can't actually calculate it. So each module can construct an infinite number of different patterns of neural activity. Thus, it can construct a nearly infinite number of different perceptions of the world. Now, each module is going to analyze a very small part of our visual field. So each module corresponds to just a tiny fraction of what we're looking at. And by combining the activity of all the different modules, we can then determine what are the very fine details of all of our visual field. Does it respond to, does this portion of our visual field contain a line with, that's horizontal, that's vertical, that's 30 degrees? Is it red? Is it blue? Is it green? Is it the same in one eye versus the other? Because most of the neurons are going to respond to activity in both the left and right eye. They're binocular, but not every neuron. Some neurons will only respond to input from the left eye, and some will only respond to input from the right eye. This is what we call ocular dominance, and it's conserved within each column because as you move down the column, you're getting essentially the same input, either from the left eye or the right eye, and then in between these there's a mixing where the inputs get put together and you have binocular vision. So we can determine then within each module what are the fine details. We don't really construct a perception. We don't know what we're looking at. We'll see the nitty-gritty. We'll see the lines, the edges, the corners, the colors, the general depth, but we won't actually know what we're looking at until we move beyond our primary visual cortex. So it's only in the associational cortices that we then construct our perceptions. So what do we see? So the striate cortex is going to project to the extrastriate cortex. So we'll go from V1 to V2. There are a number of different visual cortices. V2 is going to be the next step. Here we're still keeping our color input separate, so the blobs are going to project to what we call thin stripes, and the interblobs will project to pale stripes and thick stripes. This is again looking at a cytochrome oxidase stain. All right, same image as before, but now if we look in V2, you'll see that there are some thin stripes that are cytochrome oxidase positive, there are some thick stripes that are cytochrome oxidase positive, and then there are pale stripes that stain very dimly for cytochrome oxidase. It looks like there's some divergence here, where the interblob is splitting its signal, and it kind of is, but there's actually a lot of convergence in V2. It's the case that neurons in V2 receive inputs from multiple V1 neurons. So what we're doing is integrating information. So a number of neurons from V1 will feed into V2, so we can construct a more detailed understanding of what's going on. Each individual neuron will have a less detailed understanding of the nitty-gritty of our visual field, but they'll start to construct ideas about what we're looking at. Okay, so if we look at the cartoon on the right here, this helps us here. On the top, we're just tracking the visual input. We start at the eyes, we go to the lateral geniculate nucleus, then we go to V1, we pick out the nitty-gritty, and now we're going to go through the associational cortices, V2, V4, and then beyond there. So in V2, now if we look at the bottom, we're going to start at the very bottom of this cartoon and work our way up. In V1, we're just determining what's the angle of the line. Does it stop here? Is it an edge? What's the color? But we don't put together any relationships between all these different lines. It's just a collection of different lines of different angles and different colors. The relationships get constructed in V2. So we take all the different lines and edges, and if we happen to have 4 lines that connect in a way that creates a square, well, now we can start to detect that in V2. So here is where we start to understand the spatial relationships between all these different lines and edges that we determined in V1. Moving up further within the extrastriate cortex to V4, then we look at all the different shapes in our visual field, and we can start to construct objects. So what am I looking at? You know, if you see an octagon that's red with some letters in it, you can start to understand what this is. It's a stop sign. Of course, we're going to get a more detailed understanding of this in the next steps, where we're going to analyze either where or what something is. So once we leave the extrastriate cortex, we're going to go to 2 different locations. There's going to be what we call the dorsal stream, where we move dorsally, we move upward into the parietal cortex, and that's where we determine where an object is. Or we can move ventrally. We move down into the inferior portions of the temporal cortex to determine what an object is. Okay, so we've left the extrastriate cortex. We now have some understanding of what shapes and objects we're seeing. If we want to determine where something is, that's going to be determined in the intraparietal sulcus, the IPS, and there are many different regions in the intraparietal sulcus. The lateral and ventral portions are going to be involved in controlling the saccadic eye movements and maintaining visual attention. So they'll receive input there, you can see from V4. So this is telling the intraparietal sulcus what we're looking at, what are the objects, what are the shapes that we're seeing. There's also input from the frontal cortex, where we have plans about our motor activity. So here in the dorsal stream, we can take our visual input and use that to guide our motor plans. So we'll receive input of how we're moving our eyes or our hands, and how we should modify those movements based on our visual input. Okay, so the lateral and ventral portions are going to control the movement of our eyes and where we're attending to based on where the objects lie in our visual field. So based on input from the extrastriate cortex, our interparietal sulcus can help keep our motor plans in check so that we move our eyes appropriately. We can also control the movement of our hands and pointing with the ventral, medial, and anterior interparietal sulci. Then the depth perception really comes from activity in the caudal interparietal sulcus. This is really where we determine is something in front or behind of where I'm looking based on the retinal disparity. In order to determine what something is, that's going to be carried out in a variety of different specialized regions. So you can think of these as modules as well if you'd like. So the visual associational cortex is going to have different regions that respond to different information. So we have the lateral occipital complex. This is part of the extrastriate cortex. It's going to respond whenever we're looking at specific objects or shapes. So different neurons within the lateral occipital complex are going to respond to distinct objects and shapes, whereas some neurons might respond to a square or others to a triangle, for example, or specific objects like looking at a cat. The fusiform face area is somewhat of a misnomer. This is a part of the inferior temporal lobe, so this is in that ventral stream. This is a region that in most people is going to respond very strongly to faces, but really its job is to detect complex objects that require some sort of familiarity to recognize it. If we look at the activity in the fusiform face area, that's what's shown in the upper portion of this illustration. The fusiform face area is outlined in black in these 6 different regions. It's showing us, this is a view of the brain from the bottom. So we're looking at the inferior temporal lobe, and the amount of activity is shown with shades of yellow, where the orange is not quite as active and the yellow is very, very active. You can see that there's still some activity when people are looking at birds, flowers, houses, guitars, and cars, but the strongest activation occurs when they look at faces. And this is really because faces are complex objects that are very, very important to us. We attend to faces so that we know who we're looking at, and so we can recognize people who matter to us. But this is learned. This is not innate. We're not born being able to just recognize faces very well. If you compare the activation of the fusiform face area in children and adults, that's shown on the bottom, you can see that the fusiform face area becomes responsive to faces because we attend to them, because we deem them important. We learn how to recognize faces, and thus the fusiform face area becomes responsive to them. Really, it shouldn't be called the face area, it should be called the complex object area. But because faces are so important to humans, it naturally adopts this role. But it doesn't only respond to faces. It responds to any complex object. So just as we have a so-called face area, there's also a body area that tends to respond to parts of the body other than the face. This is not in the inferior temporal lobe. This is in the lateral portions of the occipital temporal cortex, so somewhere between our extrastriate cortex and the ventral stream there. And then we'll have regions of the brain that respond to particular places or scenes. They'll be active when you're in your room versus in your dining room, for example. And this is, of course, near the hippocampus, because the hippocampus is very important in determining where we are. The perception of motion is carried out in an area called V5, so this is still a part of the extrastriate cortex. This actually happens very quickly. That's because of the inputs that are going to V5. So it's going to receive input from V1, so the primary visual cortex, as well as other associational regions, but it's also going to get some immediate input from the superior colliculi, which you should remember as being a part of those visual reflexes. So V5 is going to get very fast input to allow it to respond rapidly to visual input. This way, if something is moving toward us, we're aware of that before we really know what it is. If someone throws a ball, you're not ready for it, and you sense that motion of something coming toward you, you can quickly respond to it. The motion is going to be determined based on the size of an object. So if it's getting larger, it's probably coming close to you. The objects will change shape to some degree as they move around, and then the location in our visual field. If it's coming from the periphery toward the center, then it's probably moving toward you. It's moving from left to right. Now, of course, every time you move your head or your eyes, everything in the world moves relative to them. So the neurons in V5 also receive input based on the movement of our eyes as well as our head. They'll receive input from the vestibular system so that we can compensate for these movements. So the neurons in V5 know which direction our head and eyes are moving, and as such, this can help cancel out that perceived movement. So even though we've turned our head and it looks like everything is moved, we're aware that we've moved our head, and so we actually perceive it as nothing moving except for our head. Only the motion that isn't consistent with what our head has done will be perceived as motion. So if an object is moving, you can still move your eyes about, and you'll perceive that object as moving. So this is essentially just internal circuitry so that V5 is aware of how our head and eyes are moving and how all the objects that are stationary should also be moving. So there's a lot of circuitry here. There's a lot going on if we're going to make sense of the world, and damage to these different modules that I've brought up here are going to lead to different types of visual agnosias or disturbances in vision. Probably one of the strangest would be prosopagnosia, that'd be the inability to recognize faces. You might think that this is caused strictly by damage to the fusiform face area, but it's not, because the recognition of faces doesn't really only occur there, it occurs outside of it as well, and the fusiform face area is responsible for a number of objects besides faces. It's anything complex. So prosopagnosia is an odd one, and it's very specific. It's just the inability to recognize faces. It's described as looking at a face is kind of like looking at someone's elbow. You know, if you look at a series of elbows, it's kind of hard to pick out who's who. Then there's acantopsia, where you no longer perceive movement. The world is kind of a series of still images, so it's very difficult to navigate the world without seeing the movement go on. Color vision is, of course, constructed in our minds, in our brains, and so damage to different brain regions can lead to what we call cerebral chromatopsia, or color blindness that's not the result of some sort of mutation in our photoreceptors. We pick up the colors fine in our retina, but we're not able to make sense of them appropriately. Some people do indeed see the world as strictly black and white because of specific types of brain damage. So here we're going to see a woman who has prosopagnosia and hear a bit about her story.
Your mouth is getting tired.
Meet Terry Sweeney, a professional photographer. She was a firefighter until an injury damaged part of her social brain, the circuitry she uses to recognize faces. The disorder is called prosopagnosia. For example, the person Terry has been photographing here is her mother. Now, because of her injury, Terry can't recognize any face.
Make me smile.
Not even her own mother's.
I'm really grateful you don't drink a lot of coffee.
In this test, Terry takes a Polaroid picture of her mother.
Terry, you just took a picture of your mother. Yeah. And I want you to take a look at these and see if any of them are the picture you took or not. They may.
Now, Massachusetts General Hospital researcher Dr. Nancy Etkoff shows Terry several Polaroids with only faces showing and asks Terry to pick out her mother.
Okay, if you had to guess, which looks to you the most like your mother? Just looking now, you can't tell which one of these. None of these really look like your mother to you.
No.
Okay.
If you just cover up the face and I just look at the clothes, I can tell you.
Okay, do you want me to uncover them? Okay, why don't I do that? I'll uncover them so you can see what they're wearing and other parts of the picture.
And see, that's her, because the pink and purple shirt.
The shirt. You recognize the shirt.
Okay, hopefully that video played at your end. So you can see that without the ability to recognize faces, you have to rely on other cues, and life becomes just a little bit more complicated as a result. Alright, I hope you'll email me any questions you have on this material, and I look forward to seeing you for the next lecture.